everyone, I'm Liya Mooradian, a rising junior studying genetics and science communication at Iowa State University of Science and Technology. I'm from Massachusetts, but I had the honor and distinct pleasure of working at the Children's Hospital of Philadelphia and the University of Pennsylvania in Dr. Li-Ming Pei's lab for the last 10 weeks. I worked on a couple projects while there, including the procurement of human heart samples, and my HuBMAP project is single-cell genomics guided understanding of human cardiac biology and disease. So to begin, I wanted to describe a very exciting experience I was able to have. And disclaimer, I do have a picture of an organ on the next slide. Since the Children's Hospital of Philadelphia is consistently ranked among the best of children's hospitals in the nation, according to US News and World Report, they interact with many patients and have lots of resources. And so one particular resource is the ability to procure human hearts that qualified for organ donation and transplantation, but were unable to be matched with the recipient. And so procurement of human hearts from these donors and performing single-cell sequencing proves important to establishing a baseline for defining what a normal human heart would look like. Listed here on the left, we have some different factors of how we obtain these hearts with informed consent, as well as consideration for the inclusion of diversity, whether that be gender, age, ethnicity, race, etc. We can secure these samples with consistent protocols for collection. And so our protocol is based on metabolomic data and involves immediate antegrade perfusion, storing in cold cardioplegia and paraformaldehyde, and snap freezing in either dry ice or liquid nitrogen to be fixed within 4 hours. So here's a picture of me last week in my second heart collection. I was very lucky that I was able to attend and learn from not only one, but 2 heart procurements, which is almost unheard of in the short time that I was at CHOP. And they both happened in the middle of the day, which I'm told is also quite rare because they usually happen at 3 or 4 in the morning. So we were very lucky in that sense. And we procure samples from the right atrium, the right ventricle, left atrium, left ventricle, aortic root, and the septum. And here on the right is pictured in dry ice for transport. So once we have these samples, we're able to perform single-cell multiomics procedures, and I'm sure you're all familiar with this standard workflow of how this technology works, as shown here. And so we're able to have our barcoded DNA and RNA go through preamplification to eventually be added to single-cell ATAC library plus the single-cell gene expression library. And here's the output for the single nucleus RNA sequencing of human hearts from 2 collections. The data is filtered using Harmony integration, and the HuBMAP on the right shows where the cells are in respect to the 4 chambers of the heart, while the left shows what types of cells can be identified. You can see that there are B cells, macrophages, proliferating cells, parasites, neurocytes, neuronal cells, lymphatic endothelial cells, atrium cardiomyocytes, fibroblasts, T cells, and some adipocytes. And it is with this baseline information that we can further apply it in experiments. And you'll see that we actually focus on fibroblasts. And so when we're not collecting hearts, we're working on a different but related project. And that is the Fontan Liver Project. So for some background information, the Fontan operation is a surgical technique used for individuals with single ventricle congenital heart disease. It is an open heart surgery that these patients receive between 18 to 36 months old. And this congenital heart defect afflicts 2 to 3 infants per 10,000 live births. The genetic cause of most patients' disease and heart defect is unknown, unfortunately. And as shown in the diagram, the left ventricle is not working. So surgery is used to connect the superior vena cava and inferior vena cava directly to the pulmonary artery using a conduit or a tube. And this restores the right ventricle to pumping blood to the lungs. and the left to pumping blood throughout the body. And it is crucial to repair this life-threatening issue. If these patients did not undergo this procedure, these children would die. And so luckily, when these children do get this surgery, there's an 80% chance of survival to adulthood. And that brings us to the finding that nearly 100% of Fontan patients develop Fontan-associated liver disease, or FALD, in adolescence. And the progression pathway of this related disease involves hepatic fibrosis, liver cirrhosis, and hepatocellular carcinomas, which will eventually require a liver transplant. The molecular mechanism, however, is unknown, but current hypotheses point to central venous hypertension, lymphatic congestion, and overall low cardiac output. So snRNA ATAC-seq was used to compare single-cell landscapes of normal and Fontan livers, investigating differentially expressed genes and biological changes in FALD proved critical. And so this was done by looking at expressed genes in all the cell types shown. Central hepatocytes, shown here as a reddish orange, have the most upregulated and downregulated genes in our findings. So armed with this information, we can look at the biological changes and note that metabolic processes in FALD patients are severely negatively impacted. And this leads to the conclusion that FALD is not only a fibrotic disease, but also a metabolic disease. And using that obtained data, we're also able to find candidate receptors from cell-to-cell signaling analysis. And to do this, we identified intercellular signaling pathways to activate HSC target receptors. HSC are hepatic stellate cells and are a type of fibroblast that are essentially key players in the development of the fibrosis that characterizes FALD, which is why we focus on these central hepatocytes. And the biggest change is between the central hepatocytes. So our hypothesis is that the pathogenesis involved profound remodeling of CHEP and relaying signals to the HSC to promote fibroblast activation, which then translate to our ligand receptor pair. So in figures A and B, the cell-to-cell interaction within Fontan livers is isolated to focus on CHEP and HSC cells. And then the table shows the ranking of different CHEP ligands and HSC receptor pairs. And so using this, we worked with inhib A, B, and C. And all this gives us the question that I most worked on during my internship, and that is, from a molecular mechanistic point of view, how are these ligands upregulated? And so we took a look at potential transcription factors, and we're able to use ATAC data to find where the enhancer and promoter are, and then transcription, direct binding, and in vitro experiments was to follow. So here are our data for the expression of these genes within the control groups versus the Fontan experimental group. We can see that inhib A, B, and C are highly upregulated in Fontan compared to the control. And on the previous slide, we were looking at those cell-to-cell signaling candidates. And here we are trying to understand how those are over-secreted ligands are regulated. And here we have the SNAP gene flow chart for how we were able to understand how these over-secreted ligands are regulated. And the colored markers are ZNF148 and NRF1, which are transcription factors that we wanted to see how they regulate the expression of inhib A and B. And then for our next steps, here's a model of FALD pathogenesis. And these would be the next steps and avenues to explore. The top left corner in this little circle of what would be a CHEP nucleus would be the investigating using NFYC, ZNF148, PPAR alpha, and PPAR gamma genes. So working on such a spanning research project allowed me to gain experience with a variety of experimental techniques. I had quite a bit of research experience when I arrived at CHOP, but I gained proficiency in unfamiliar techniques like cloning, transfection, sample procurement, single cell data analysis, flow cytometry, and immunohistochemistry. And then here are just some snapshots of my time here in Philly. To the far left is my PI, Dr. Pei, and myself, as well as my lab mentor, Po, a UPenn grad student. And I felt really welcome in the lab during my time here and was even included in the lab photos in the top middle. Plus, I had my very own desk. And along with gaining lab skills, I also got to attend different lectures by renowned scientists and make valuable connections. These are all the organizations and individuals that I'm grateful to for my time in the Pei Lab and for any aid with my research project. I'd like to especially mention again my mentor, Po Hu, as well as my PI, Dr. Li Ming Pei, as well as our HuBMAP program, CHOP, UPenn, and the NIH. Lastly, connect with me on LinkedIn. Thanks for listening, and please feel free to ask questions now or send me a message later.
Awesome.